Okay, so let's talk about transcription. Before we even start, I want to put the whole sequence of gene expression events here on the left so we can refer to it all the time. Okay, so remember we start with the DNA. That's our recipe book. Certain genes, not all of them, but specific genes, get transcribed. After the transcription, we'll get to that part. In some organisms, so you get RNA. In some organisms, RNA can undergo the process that is called splicing. And then spliced RNA can be used in the process of translation as a template for the synthesis of proteins. Okay? So let's start with the process of transcription. We have a DNA molecule. Okay, so this is your DNA molecule. And the gene, the actual sequence of the gene starts, let's say here. So that's nucleotide number 1. Now, turns out, in front of a gene or a cluster of genes, DNA has certain very specific regulatory element that's called promoter. So what is promoter? Promoter is like a name of the recipe in your recipe book. So imagine that you want to make a peach cobbler, okay? You're not just going through each page, right? What you do is you look at the content page, right? And you see peach cobbler, page 52. You go there, a peach cobbler on top. Does it tell you how to make it? No. All it tells you is the recipe for peach cobbler is here. Does that make sense? So the protein, the enzyme called RNA polymerase, binds to the promoter. Okay? So now RNA polymerase moves to the beginning of the gene and it unzips DNA molecule. This is obviously imperative. It has to separate the strands. So this is your big S RNA polymerase. Okay, it's moving to the right. Okay, moving this way. And RNA polymerase basically reads the sequence of nucleotides on the DNA and inserts matching nucleotides in the RNA. And this is your RNA molecule right here. Okay. That makes sense. Now, one interesting look at few interesting details. So, well, first of all, this one is called a transcription bubble. That's an official term. And basically, what we're talking about here, look, this, the start of transcription, the very first baby steps of transcription, is initiation. The process of RNA synthesis is elongation, because RNA gets elongated. And then eventually termination comes. We're going to talk about termination separately, so that's RNA. But what I want to highlight for you, look at this. How many strands are actually being read and used as a template? Just one. So this strand here is called template. Very creative name. This strand is called non-template. Does that make sense? So there is a really big temptation here. Can we just ditch non-template strand? Can we just well, interestingly, we really can't. Like, what I'm trying to make a following point here. Turns out that for transcription to happen, DNA must be double-stranded. You see what I'm saying? That's it. It must be double-stranded. Not single-stranded, double-stranded. Although we're not going to use one strand at all, it's got to be there. So we take a double-stranded DNA molecule, read it using RNA polymerase, producing single-stranded RNA, messenger RNA. Are we clear about that? Any questions on this part? Now, is this RNA, and yeah, termination. What happens during termination? At the end of the gene, there are usually terminal repeats. So RNA polymerase arrives to this terminal repeat, starts to stutter. The complex loses the stability, the enzyme falls off. That's it. The replication, transcription is done. Good? So what's the next step? The next step, if you are eukaryote or a virus, there are some viruses that use it, it's called splicing. So eukaryotic RNA gets spliced, and some viral RNAs. So what happens during splicing? A lot. Thomas Cech got the Nobel Prize for it. But basically, you've got an RNA molecule, okay? So I've got RNA right here, right after the transcription. But it turns out that RNA contains both meaningful fragments and meaningless fragments, which probably shouldn't call meaningless, but who cares? So meaningful fragments called exons, and meaningless fragments called introns. What does that mean? Meaningful versus meaningless. So DNA in virtually all eukaryotes contains the parts that encode for the protein and parts that do not. Again, going back to the cookbook analogy, imagine that you start reading the recipe for the peach cobbler, and the actual recipe for the peach cobbler is interspersed with the instructions how to cook crystal meth. If you will just follow through, the result is going to be really interesting, right? So I have to cut out the parts that make no sense and stitch back the parts that do. Are you following me? So the DNA that doesn't encode and kind of meaningless is often referred to as junk DNA, which is not really a good name, but it's okay. Good? So what happens during splicing? We're not going to go about molecular mechanisms, but basically introns are removed and exons are stitched together so that RNA transcript becomes a proper mRNA with exons being stitched together and introns, being thrown out. Does that make sense? So, I want you to notice one really important thing. In eukaryotes, transcription and splicing happen in the nucleus. Does that make sense? So, when we finally arrive to this point, look, when we finally arrive to the point of RNA, like proper mRNA in eukaryotes, and we're ready to make protein, we need to transport that RNA from the nucleus after all of this into the cytoplasm or endoplasmic reticulum to the ribosomes. Does that make sense? In prokaryotes, you don't have that barrier in the form of nuclear membrane. You with me? So basically, if this is a bacterial DNA, this is the bacterial RNA. Remember, bacterial DNA is right there in the cytoplasm. Does that make sense? So that RNA is sticking out. And it's ready to roll. It's ready to be translated right away. So it illustrates first big difference between the gene expression in prokaryotes and eukaryotes. In prokaryotes, you have a spatial barrier. You have to transport RNA from a nucleus into the endoplasmic reticulum or cytoplasm. Does that make sense? In prokaryotes, you don't have that. There's no membrane separating DNA from the rest of the cell. And second big barrier in eukaryotes is splicing itself. In eukaryotes, RNA has to be processed. Does that make sense? In prokaryotes, when it comes out of the transcription bubble, it's ready to go. There's no splicing, there's no RNA processing. It's good. Ribosomes can jump on it. Does that make sense? And this is actually what they do. That's going to be kind of a little concluding remark before we move on to translation. This is what they do. In prokaryotes, in bacteria, ribosomes will jump on the RNA while it is being synthesized. Does that make sense? So in this illustration of mine, RNA, the transcription is not finished. It's just a part of RNA, but it's already being used as a template for the protein synthesis. Does that make sense? So basically, I usually compare it to what we have now in the internet age. How would you read the book before? A person, an author, would write a novel, finish it, give it to an editor, okay? Then give it to a publisher, and give it to the editor and the publisher, and only after that, that novel will get to the reader. Okay, so you need to process. Now what you can do, you can start writing novel and just publish it online in your blog or whatever. Okay, you haven't finished the book yet. You haven't finished writing the book yet. But you already have people who read it as you are writing it. That's your prokaryotes. That makes sense? That's kind of a big difference. We good so far? Now that another difference between prokaryotes and eukaryotes in terms of the transcription is number of genes in a single transcript. So if you look at prokaryotes, imagine that you have DNA with genes 1, 2, and 3. When this DNA is transcribed into RNA, it can be a single molecule of RNA with genes 1, 2, and 3. This is called polycistronic transcript. In eukaryotes, same arrangement, 1, 2, 3 genes in the DNA. When they're going to be transcribed into RNA, you're going to get gene 1, gene 2, and gene 3 as separate RNA molecules. And it's called monocistronic transcript. So to kind of summarize the differences between eukaryotes and prokaryotes here. In eukaryotes you have spatial problem, right? You have that nuclear membrane that your RNA has to cross. And you have time problem. You got to go through the RNA modification before that RNA can be used for protein synthesis. In prokaryotes, you have neither of those. In prokaryotes, in fact, transcription and translation occur almost simultaneously, almost at the same time. At some point, they do happen at the same time. Are you with me? And the transcripts in prokaryotes, polycistronic, in eukaryotes, monocistronic. Any questions on this one? Okay. We're going to talk about translation. I'm not going to do a separate video on that because, well, we'll see. No, actually, because we need to talk about something else.